Today marks the achievement of your undergraduate career. You've learned a lot, you've worked hard, you've been exposed to many new ideas, many new people, many new things. I think you will look back upon these years as for many, many years to come. And so it is with pride and with pleasure and with a little bit of fun that we celebrate today your achievement, and I want to welcome you to the Society of Educated Women.
Okay, so what I'd like to do is talk a little bit about the major and about what your students have done. First of all, you should all be parents. You should be very, very proud of your students. This is one of the most difficult majors on campus. And as we'll see in a second, your students have done extremely well, exceedingly well, and have accomplished quite a bit. Us as professors, why are we interested in research? Why do we have research labs and carry out research? And what is, why is that interesting to us? And so what we wanted to do is to sort of expose students to that, joy of discovery, of figuring things out. And so we thought that the best way to learn science is to actually have the students do science. And this really isn't a brilliant principle, right? If you think about it, how do you learn how to play baseball? You don't read a book, you actually get out there and practice. Learn how to play piano, the same thing, it's through practice. And so we want students to learn how to do science by actually conducting research projects. So every student in the MBB major conducts an independent research project with a faculty member on campus. Okay, so what have they done? Well, we have a relatively small major on campus. And there's benefit, as you'll see down the road. We'll be over by 11 o'clock. Right? 21 but of these 27 students, we have one of the highest GPAs on campus, 3.7 average overall. We have 21 general honors students. Okay, that is the highest percentage on campus. Okay, very, very impressive. In addition to this very heavy major, we have 3 students who have done double majors, okay, and 18 who have done minors. And again, they're not required to do minors because this major is so credit intensive. So this is really quite an accomplishment for them. The proteins using the single protein production system in E. coli. So this is a gel showing some of his proteins that he's produced. Thank you. So Stephen's work was heme oxygenase 1-dependent central cardiorespiratory adaptations to chronic intermittent hypoxia in mice. Fragment screening of the influenza type A non-structural protein 1, NS1A effector domain.
I think the future.
He has project engineering thermostable alpha amylase and higher plants for the production of biofuel. Analogs and health span of C. elegans, making worms live longer. 3' UTR on the survival and metabolism of Drosophila melanoic acid. 5, a novel gene functioning in Drosophila oogenesis. I want to point out, she did these figures. Very, very impressive. She's got a career graphic. Project is remodeling Rhodobacter supporites photosynthetic units after growth condition transition. Albumin nanoparticles for the targeted delivery of therapeutic U1 adapters. Project was decathlonic acids from chicory inhibit hepatic glucose production in vitro. The project is functional analysis of conserved residues in fission yeast of tropomyosin. Observing the effects of chemoheterotrophic growth and the transition from high light to low light on the assembly of the photosynthetic apparatus of the purple bacterium Rhodobacter subfloroides. Is the induction of pluripotency by direct transduction of proteins from retroviral gag polyprotein using Maloney-Murine leukemia virus. Gamma tocopherol and BPA on NNK-induced malignant transformation of human mammary epithelial cells. The protective mechanism is resveratrol against X-ray-induced DNA damage in mouse embryonic stem cells. Investigation of the sensitivity of 2 prostate cancer cell lines to acute and chronic heat shock with a consideration of the role of EIF5A depletion. Thanks. Chemoheterotrophic in the high and light to low light growth conditions on the assembly of photosynthetic apparatus and Rhodobacter subfloroides. With the experimentation and analysis of different ratio aggregates of alpha and beta pancreatic isolate. Rescuing MLV P12 mutants with DNA tethering domains. Is categorizing an antibody response against HER2 overexpressing breast cancer. The cellular pathway of BDNF promoted increases in dendrite branching. First and fourth place is George Ganem. Next was for third place is Leah Weinstein. Come on up. And Leah's project was the categorizing the antibody response. And we have a tie for first place. I'll first call up Eve Hardy. Come on up. 5, a novel gene functioning in Drosophila oogenesis. Okay. Also tied for first place, Melissa Pamuel. For research on functional analysis of conserved residues in the fission yeast tropomyosin. So I have 2 last things to ask you undergraduates. All right. First of all, well, not first of all, but I want you all to have a great career. I want you to think about the things you've learned, you're discovering as an undergraduate, and to keep that throughout your life. Right? Our goal is to have lifelong learners, right? Constantly investigating, thinking, evolving. So we want you to keep that spirit that you had here throughout your life in everything you do. Second thing is we want you to stay in touch, right? We want to hear your successes. We also want to hear your impressions about the MBB major down the road. I know I have got one last request. And most importantly, I want you all to thank your parents, okay? They've sacrificed a lot. They paid a lot and all the rest. I want you to all thank them for your 4 years here, for their support, and especially your mother.